Okay, so it's 12 noon. Good afternoon, everyone. Thanks for joining us for Medical Grand Rounds. Before we get started, just a reminder to please answer an email that you will receive after the presentation. This will be a survey which will let us know that you were present at today's rounds. And I would also ask you to please enter your questions in the chat at the end of the presentation. Our presenter today comes to us courtesy of the Division of ID, and I'm going to ask the Chief of ID, Marcel Baer, to please introduce our speaker today. Marcel.
Thank you, Nadia, and thank you everybody for attending. I was sent via email a Vinh bio sketch, and as many of you know, when you introduce somebody, you're supposed to read a really long and detailed bio sketch, and they're supposed to gush and say, that's so wonderful of you. And I'd like to say that Don's biosketch was really boring, and it didn't describe him in any detail, and it didn't provide half the flavor of the Don Vinh who I know. Don Vinh, so I'm not reading it. Don Vinh is known to some people in the hospital as our Dr. House because he solves completely bizarre medical mysteries, many times decades after the onset of disease. He's known to you in the media for being one of the strongest and most consistent media presence during COVID. He's known at my twins' school as the medical advisor who kept COVID at bay at the school, and he's got a lot of community activities, and he has to be thanked, and you know, it's remarkable how much he's doing. And he's also one of my teachers, and he teaches that if you study something in depth, you can, in few patients, you can learn generalizable messages. And he once told me that if you understand why one person has a defective Krebs cycle, there are universal lessons about the Krebs cycle. And we've learned through COVID that some of the lessons from genetic studies of infectious diseases also have lessons beyond just genetic defects in how we handle infectious diseases. So he's one of the people who's taught me consistently that infectious diseases are more than microbes. They're microbes that interact with the host. And there are so many lessons, and he's going to share some of them with us. And I'm sure there are many more lessons to come. Thank you for speaking with us, Don.
Oh, well, thank you very much, Marcel, for that very flattering introduction. Yes, that is much better than that boring dating bio sketch that I gave you, which probably would have attracted nobody. I'm going to share my screen now. So I'm going to ask the generic Zoom questions. Can you hear me? And can you see me?
We can hear you and we can see you, Don. Thank you.
Great. So the title of my talk is, as it says here, Infectious Disease or Immune Deficiency, 6 of one or half a dozen of the other. Because I'm actually not sure if there's a distinction between these two, what have been typically silos. So the agenda for today will be as follows. We want to understand what an inborn error of immunity is. I'm hoping that we can recognize errors of immunity to the field of both human biology, so at the scientific level, and precision medicine at the clinical level. And I'm hoping to sort of stimulate or help others appreciate, especially those who may be unsure if they want to pursue a career in scientific research, to appreciate the breadth of diseases that are waiting to be discovered in medicine by studying a few patients really well. So for those of you who don't know, every Friday afternoon, we have ID case rounds, and they are weird and wonderful. And by the way, weird and wonderful applies not just to the cases, but obviously to the faculty as well. But that's a different, that's for the psychiatry rounds another day. And every time we have case rounds, we always talk about, hey, there's this weird case where this patient has a bug, and we know the bug and we know the drug, but we're giving it and it just doesn't work. And what's going on? So that's unusual, right? And usually refractory to traditional approaches, or it's unusually severe, like this is TB that's miliary or extrapulmonary, or this is just unusual. It's caused by a bug that nobody in the room can even pronounce. And the question has always been, of course, we hem and we haw for that hour, and then we sort of, close the books and go on our merry way. But I think these cases are actually opportunities for us to learn about infectious diseases. What do I mean by that is that the classical approach for infectious diseases is based on the germ theory of disease, pioneered by obviously some of the greatest microbiologists of our time, including Pasteur and Koch and a bunch of others whose names live in the bacteria that we speak. And of course, the idea behind the germ theory is that a bacterium, at least at the time, right, it was restricted to bacteria. A bacterium causes infectious diseases. This obviously was the guiding theory for medicine at that time and since then. But there's been a parallel theory that perhaps hasn't garnered as much attention, pioneered by Charles Nicolle in the early 1900s, for which he got the Nobel Prize in 1928, which is called the Infection inapparente. And what he noticed is that a bacterium doesn't cause an infectious disease. A bacterium causes a spectrum of manifestations. And that spectrum can look like this. Any one person, when you're talking about a microbe, could be potentially asymptomatic or colonized, depending on the microbe, or they could develop mild disease, or they could develop severe disease. So there's this natural variability in susceptibility to developing infectious diseases. Obviously, in the hospital, we're very biased to those that are severe and sometimes mild. We don't think about the asymptomatic or the colonized, but that's a huge piece of the puzzle that we're missing. So why make this distinction? Well, I think it changes our thought process, right? If we think or focus exclusively on germ theory of disease, then by definition, we will develop pathogen targeting antimicrobial therapy. And that is essentially the field of infectious diseases. What new antimicrobial can we develop and use? But if we also focus perhaps on the host and what's not working in the host, then perhaps we can develop host-directed pro-macrobial therapy instead of microbial, the big guy instead of the little guy. And so this goes back to a point that I had learned from Henry Kunkel. I didn't learn from him directly. It was very inspiring to go to Rockefeller University and see a statue in his honor. For those of you who don't know, Henry Kunkel is both a clinician and scientist. Here are some of his accolades listed here. But the key point that he actually emphasized was not the actual work that he's known for, but more that strategy, right? Which is you need to be able to study a few patients well. And so I've actually taken this as sort of my mantra and developed a program to this effect. Because when you look at weird or extreme phenotypes of infectious diseases, the question is, are these due to an inborn error of immunity? Now, we've heard of inborn errors of metabolism, but now we're going to talk about inborn errors of immunity, which really comes down to defined as single gene penetrant defects of immunity. Sometimes you'll hear the word Mendelian, although it's not strictly speaking a Mendelian phenomenon. But the point is that a single gene that that's obviously defective penetrates to the clinical phenotype and causes an immune defect that then manifests itself as, at least in our field, infectious diseases. So then the hypothesis is, are these inborn errors of immunity, these EIEs, do they underlie infectious diseases in some or maybe many or maybe in all of the patients that we see? And I think at least in some of them they do, and I'm hoping to give you some examples of what we've been able to study so far. So how do we study these people? Well, first and foremost, we have a whole plethora of these people every day. I'm on service this week, and I can tell you there are people who have these. They have infections that are either recurrent. In other words, the person who keeps getting sinusitis and pneumonias or recurrent skin infections or recurrent abscesses, or they're refractory. They have bug X, we've given drug X, bug X should be covered by drug X, and it just doesn't work. The person's not getting better. Or it's severe. The person who has group A strep in the throat and has no symptoms whatsoever may be sitting next to somebody who has group A strep eating away at his leg. And so the question, of course, is, well, what's the difference between these two? And it's not entirely microbial. So how do we study this? Well, you create a platform by which you can study this. You create a laboratory program, a research program, a center, if you will. And that's what we've done, where we have two major avenues. First, we have those that are pathogen-directed, and we study it for host genetic defects. In other words, these people have a problematic infection to a singular microbe, but it's quite problematic. And maybe by teasing this out and dissecting it further, we can understand human immunobiology. And the other pathway, which is complementary, are those who don't have a problem to a microbe, but they have a general problem. You can tell by the constellation of features, and I'll give you some examples of that. And again, we want to study these people. So that's the framework. And now the question is, well, that's nice. That's how you do it, scientifically. But how do you actually approach this from a clinical scientific perspective? Well, at the start of the day, I'm a physician, so I want to know what is the clinical phenotype. I want to know the disease. I want to define it, and I want to understand the natural history of that disease as much as possible. I want to get to genotype. Obviously, these are genetic disorders of immunity, so we need to find the gene, find the variant or the mutation, if you will, and prove that that variant is truly a mutation by proving that it's molecularly deleterious. That molecule that's deleterious has to have an impact on the cells that harbor that variant, right? So we need to have a cellular phenotype. How does that mutation affect the cellular phenotype, which then eventually leads to the clinical phenotype? We want to be able to dissect molecular pathways. We want to be able to figure out which molecules are interacting with what, which of these pathways are dysfunctional. And of course, that would be ultimately to be able to translate that into treatment. Because we can go through this exercise and be all nerdy and scientific about it, but at the end of the day, as physicians, we want to be able to take that advancing science and lead it to novel therapeutic approaches. So I know what you're thinking. You're saying, okay, he just talked about genetics and immunology and infectious diseases, and I am, petrified, if not scared, that this is going to be very boring. And I'm hoping that at the end of this presentation, you will be more rejoicing. So let me give you the first example that we've been able to achieve. This is, for those of you who don't know, this is an MRI of a brain. And for those who are not neuroradiologists, that white thing here is not normal. This is what we call patient 1, who is a mid-30-year-old person who developed seizures, was found to have this mass in his brain. There was some delay in getting it resected, but because of ongoing seizures, this was resected. Interestingly, they thought that probably this was going to be some sort of tumor, maybe malignancy, maybe benign, but some sort of a tumor. But when they looked under the microscope, they saw this. This is a picture of the histopathology. Thank you to Marie-Christine Guillot in neuropathology for these images. And what you can see here is that on a PAS, or periodic acid shift stain, these purple things are actually fungus. And when it was cultured, the tissue actually yielded Candida albicans. Now, just to put in perspective, we're all colonized with Candida albicans. In some people can cause, denture stomatitis or diaper rash or other types of mucosal candidiasis. But we all agree that having Candida in the brain, especially in the absence of any medical history, risk factors, or interventions, is just bizarre. So now this bizarre case would have been sort of dismissed, except for the fact that when you start looking for bizarre, you start finding bizarre. This is P2, second patient, who also was in his mid-30s when he had seizures and had this imaging, and these soap bubble lesions were interpreted as being glioblastoma. Unfortunately, because of ongoing seizures, he ultimately needed a resection, and fortunately for him, on the histopath, there was no glioblastoma, there was no cancer, there was no mass, it was pure fungus. It was, in fact, pure Candida albicans, as in P1. P3 presented to Lunetary New Look with a problem with her vision, and the optometrist said, there's something wrong with your eye, sent her to the hospital, and she had this white cheese ball floating in her eyeball, which ultimately was aspirated and found to be albicans. When they contacted me and said, well, this is a bit weird, what do you think of it? I said, scan her brain. And lo and behold, in her brain, she had a mass lesion, which is presumed to be Candida albicans. And because she also had back pain, we did an imaging of her back, and she also had osteomyelitis. Based on the eye lesion, we treated her with therapy for Candida, and all of this improved, suggesting that these were all the same process. She actually had an identical twin who was less symptomatic, but on screening with MRI, she had what was read by the neuroradiologist, not by me, that she had all these abnormal lesions of an old or remote process that was probably healed now. So the disease that we're talking about is what I've called spontaneous central nervous system candidiasis, because these people developed CNS disease with Candida albicans, and yet they had no classical risk factors for developing invasive fungal disease. So we ended up sequencing them, doing a bunch of genetic investigations, and found recessive mutations in CARD9. The first patient was homozygous for this mutation that's labeled PY91H, and the other 3 patients were compound heterozygous. I'm just going to show you an example of what we found so that you can do Sanger sequencing for the gene of interest, and you can see what a control person would look like. Here, they would have a red lesion. P2 has both a red and a blue mark in their chromatogram, but when you look at the cDNA or the gene that's expressed, it's only the mutant blue. Same thing with P3, and same thing with P4, the identical twins, suggesting that this is abnormal because when you look at the mother of P3 and P4, both genes are normally supposed to be expressed. So all P2, P3, and P4 are compound heterozygous, and that's because of something called allelic imbalance, which I won't get into. But at the end of the day, these 4 patients are transcriptionally equivalent to each other in terms of being recessive for this CARD9 mutation. So that's establishing the genotype. What's the cellular phenotype? Well, when you take cells from P1 on the top row or P2 the bottom row, and you stimulate it with all sorts of candida, whether it's a commercial candida or a proprietary candida, or even the candida that's isolated out of one of the patients, you can see in white the normal response, and you can see in gray, at least in P1 and P2, you can see even more abated, that there is an impaired response to the candida albicans in terms of the capacity to produce GM-CSF, which is granulocyte monocyte colony stimulating factor, kind of related to GCSF. And why that's important is because there's corollary data from another group, actually from several other groups, only one is shown here, is that if you take otherwise healthy people and you stimulate them to a bunch of microbial agonists individually, if you take Candida albicans, the only agonist that will stimulate GM-CSF to any robust degree is Candida albicans. And the corollary is the only way you're going to get GM-CSF produced is through using Candida albicans, suggesting that Candida albicans and GM-CSF are intimately related to each other in healthy people and are defective in these mutant patients. I'm not going to go through the molecular data, but we then found out or demonstrated that this mutant CARD9 impairs not the canonical signaling molecular pathway, but a different non-canonical signaling pathway. And at the end of the day, we want to go back and say, well, how do we translate this information? So P1, remember I told you he had recurrent seizures and he had recurrence of C. albicans despite antifungal therapy. I'm going to show you the chronology of his lumbar punctures, with blue being the CSF protein concentrations and black, the dashed lines being the CSF cell level concentration. And you can see there's always pleocytosis and elevated CSF proteins when you look at the normal levels in the dotted lines here in blue and black as indicated. Sometimes you can see a red cross, which means that C. albicans was isolated from the CSF. Now, this was occurring consistently. You can see this over a span of 4 years, and it was occurring despite being on antifungal therapies, as outlined on the top. So it's not a failure of antifungal therapy. It's not microbiological failure. It's that the disease was persisting because of immunological failure. So if his cells can't respond to Candida albicans by producing GM-CSF, could we give him GM-CSF, which actually exists to be used in humans for other reasons, and see if there's an improvement? So that's what we did. And in fact, there was not only clinical improvement, there was CSF improvement. We celebrated a little bit too early. As you can see, this was December when he had improvement. For Christmas time, I wanted to give him a break. We stopped the GM-CSF, and lo and behold, there was recurrence of his symptoms and worsening of his CSF parameters. We put him back on GM-CSF, and it's all normalized. And to this day, now 8 years later, he's well. He is married. He has a child. And not only that, but his brain works well because he stopped working for SNC-Lavalin and now works for another engineering company. So all that to say is, well, you could say, well, that's just a one-off. Who knows what that means? Well, we did the same thing with P2. P2, and remember, he's the one with the soap bubble lesions that was told it was glioblastoma. It wasn't glioblastoma when it was resected. And at this point, he was having seizures. He had the Candida albicans. He was on antifungal therapy. And despite that, things were not controlled. He was not clinically controlled. And he was deemed palliative. Remember, at 39 years old, he's deemed palliative. Now, because we were able to show that he can't produce GM-CSF in response to Candida albicans, we gave him the GM-CSF as well. We did repeat lumbar punctures, although he's from the Sherbrooke area, so we were a little bit more limited in the frequency of our investigations, but he showed the same pattern. When we gave him GM-CSF, his CSF parameters improved. Radiologically, he improved. And not only radiologically, but clinically, to the point that the Quebec government realized, wait a minute, you're still alive 6 years out, you're not palliative anymore, and they removed his palliative care privileges. So this is an example of pathogen-directed host genetic defects that we focus on. Here, I gave you an example of autosomal recessive CARD9 deficiency, which predisposes to spontaneous development of invasive fungal disease like Candida albicans. There are other fungi, but I won't get into it today. It, I think, tells us that CARD9 is critical for host defense against fungi. It also, I think, tells us that there's a cardinine GM-CSF axis that's critical for human immune response or control of at least certain fungi like Candida albicans. And now we're using this pathophysiological framework to understand invasive candidiasis in people who don't have mutations in cardinine, as well as in other fungal diseases with some collaborators across the world. I'm going to talk to you now. I'm going to switch pages and go to the other arm of our research program. We're looking for genetic defects of immunity. This patient was sent to me, thankfully, from two colleagues in gastroenterology, Talat Bessasau and Alain Biton, where this gentleman has bad Crohn's. Now, just to let you know how bad it is, there's arrows from the radiologist, and you can see donuts. And those are not donuts he ate. Those are donuts because there's so much swelling and stenosis of the luminal component of his intestines that they show up like donuts. So that's not good. And by the way, it's suffice to say, he actually doesn't respond to traditional treatments for Crohn's disease. But when you probe a little bit further, he also has bad warts. Now, how bad is it? Well, it's not just on his fingers. They're numerous on his fingers. And they're also not just on his fingers, but you can see here, they're on the base of his thumb, or on the finger on the right, they're on his ankles. In fact, I can tell you, they'll even go up to his knees. And I think we all agree that both the number and the distribution suggest that this is not usual warts. And when we do further immunological investigations, you can do what's called a T cell, B cell, and NK cell enumeration, or a TB&K. And what you can see here is that he has low lymphocytes, particularly low CD8 positive lymphocytes. So we took him when we did the whole exome sequencing on him to try to find if there was a genetic etiology that could explain this person's disease. And we did. We found a gene called Diablo. Now, there were other genes as well, but through our bioinformatics analysis, Diablo was seen as the predominant candidate. Why? Because if you look at the programmed cell death pathway as illustrated in this figure, Diablo is a nuclear encoded mitochondrial molecule that is released from the mitochondria upon stimuli for programmed cell death, but it interacts with two other molecules. This is Diablo here. It interacts with this molecule called SHIAP, which is actually already known to be a monogenic cause of Crohn's disease. And it also interacts with a molecule called RIP, which has recently also been shown to be a monogenic cause of Crohn's disease. So there's physiological homogeneity, if you will, that suggested that Diablo was the relevant gene for this patient's phenotype. And of course, because the gene is called Diablo, well, you have to name the disease. And he has diffuse enteropathy, he has verrucae, and he has impaired lymphocytes. So the disease, we've called it Devil syndrome. Now, what's the molecular investigations we did? Well, first you have to confirm the mutations. So we did. You can do that by Sanger sequencing, and we can show you here that there's a blue and a red in the P1 rows that correspond to the patient, and that he is heterozygous for this extremely rare variant. On a protein schema or cartoon figure, the mutation is indicated or at least predicted to occur where it is in red, where the arginine residue is not converted to a premature stop codon. Now, we actually have to prove that this is molecularly deleterious. So you can take immortalized cells from the patient, what we call lymphoblastoid cell lines or LCLs. And you can see if you look at P1 and as compared to healthy controls or to an unrelated immortalized cell line, you can see that the patient has truncated forms of this Diablo corresponding to the premature stop codon predicted in silico. Now, remember, this was the pathway that Diablo was supposed to be involved with. The first component you can see is that it's involved in regulating apoptosis or programmed cell death. So we want to see if that's abnormal in this patient. So you can take otherwise healthy controls, you can take people who have Crohn's disease but don't have mutations in Diablo, and you could take this patient. And then you can either not stimulate it or stimulate it with something called staurosporin that actually is known to stimulate cell death. And you can measure cell death or apoptosis through flow cytometry. And what you can see is you take healthy controls, you leave them alone. This is their basal level of cell death. You give them a cell death, stimulating agent, and cell death goes up 6.83 to 18.7. You take Crohn's disease patients who don't have mutations in Diablo, and you see you also see a corresponding increase in the amount of cell death, suggesting that this impaired cell death that we're hoping to find is in fact not a feature specific to Crohn's, but a feature specific to the patient. Because when you look at the patient, you can see that, in fact, there is no stimulation of cell death. So that tells us that the Diablo variant that we've identified has a functional consequence, which is impaired cell death. And this is just a graphic representation of that data repeated over numerous experiments. Now, if that's true, and we have to work our way a little bit more upstream than the end pathway of or the end result of apoptosis, and look at how apoptosis is stimulated, and that's through the caspase 3-7 pathway as indicated in the figure. So you can actually measure whether Diablo impairs this process. So you create, in this case, a cell line that either has the normal Diablo, what we call wild type Diablo, or the mutant Diablo, and then you either complement it with wild type Diablo, or you use a molecule called BV6, which is a known chemotherapeutic agent that sort of failed as a chemotherapeutic agent, but is in fact a Diablo mimetic. And what you can see is that, long story short in this figure, is that the mutant Diablo impairs activation of caspase 3-7 that can be rescued either by the Diablo mimetic or by putting in the Diablo wild type, suggesting in fact that this Diablo variant is a true mutation. Now, that's nice, but how does that explain the patient's Crohn's? Well, remember, the Crohn's is extremely stenotic. So maybe it's stenotic because the cells that are supposed to die are just not dying, and that leads to the stenosis. Maybe, but why is he even having Crohn's to begin with? Well, Diablo, I said, interacts with a molecule called SHIAP. And SHIAP, as indicated here, is a monogenic cause of Crohn's disease, but it also interacts with a molecule called NOD2. Now, NOD2 is actually one of the most or strongest allelic determinants of Crohn's disease, especially ileal Crohn's. And my lab happens to be next to Marcel Barre's lab, who's been studying NOD2 for a long, long time. So this is an opportunity for collaboration where we took his lab's expertise in interrogating NOD2. We then took what we call a CACO2 cell line, which is a cell line that represents intestinal epithelial cells. And there we either put the Diablo wild type or the Diablo mutant, and we interrogated this pathway's function. So what you can see here is that if you stimulate through a variety of agonists, in this case, MDP and PGN are bacterial agonists, and you use on the y-axis a readout, which is beta defensin 2, which is one of the antimicrobial peptides that our epithelium naturally releases to fight off bacteria and other microbes. You can look at what the impact of the Diablo variant does on this response. And then long story short, if you put in the wild type, the normal Diablo allele, you get high amounts of blue. That means you get a high amount of beta-2 defensin expressed. If you put in the pink, which is the mutant Diablo, you can see that this response is nearly completely abrogated, or if not severely diminished. And if you put both the wild type and the mutant together, you see that you get an intermediate response, which is, of course, probably what the patient is going on in the patient's intestines. Now, this is for one antimicrobial peptide called beta defensin 2, but you can see that with other antimicrobial peptides, in this case, HMP1, or another one called cathelicidin or LL37, you see the same pattern. The wild type induces a normal response. The mutant is what we call loss of function. And if you put the two together, you get an intermediate response that suggests what we call, at the molecular level, negative dominance or dominant negative inhibition. So this tells us that the mutant Diablo, at least in colonic intestinal epithelial cells, impairs innate immunity. You can also look at inflammatory or immunological responses using the same model, but different stimuli and different readouts. And what you can see here is if you stimulate it with IL-1 beta, remember again, if you look at the pink, that's the mutant, or if you look at the gray, that's the combination of wild type and mutants, you get more IL-23 being produced than if you had the wild type. Same thing with IL-6. If you give TNF, you get a lot of IL-6. So these two figures here are representative of the fact that not only does Diablo impair innate immunity at the top, but it also causes immune dysregulation. So if you've been reading the Crohn's literature, you would understand that these two processes, impaired innate immunity and immune dysregulation, are actually at the core of what we think causes Crohn's disease. And we think that we've been able to recapitulate those two concepts with this Diablo mutation. And as I said, the Diablo mutant protein interferes with the wild type protein, and we took a different cell line just to make sure that it wasn't inherent to the CACO2 cell line that we were using. And you can see, yes, in fact, that the mutation, because it's truncated, it actually binds to the wild type and prevents that wild type from functioning properly. Now, what about the warts, right? Because that's pretty significant. So here we created a different model, an in vitro model, where we took an immortalized keratinocyte cell line called NICS. Now, just to remind you that when you have skin, right, you have a basal layer of keratinocytes that then, sort of differentiate as they move up or outwards to the skin. And then you have a dead layer of keratinocytes at the top. And HPV, the causative agent of warts, infects the basal layer of the keratinocytes. It does not go, we think, deeper than that. And so it establishes an infection in the basal layer. And then as the keratinocytes move up, you're supposed to have programmed cell death that helps mitigate HPV infection. But HPV is known to interfere with that process. And that's why you get this overgrowth. You have impaired cell death. That overgrowth is what we call the warts. So can we replicate that at least in vitro? And what does the Diablo mutation do to that? Well, the answer is, long story short, yes. If you look at Diablo wild type, and you see that there's a certain amount of baseline apoptosis, if you infect it with an HPV-11-like particle, which is what we created in our lab, you can see that there's an increase in apoptosis of our keratinocytes with this infection with this HPV-like particle. You do that with now instead of putting in the wild type, you have a mutant molecule, and you can see what the baseline level of apoptosis is. And when you infect with HPV-11, you can see that there's an impairment of apoptosis in the keratinocytes. So we think that explains one concept, or at least one aspect of the warts. The other thing that happens, of course, is when you have warts, while lymphocytes from the dermis and the subcutaneous tissues try to get into the keratinocyte layers to try to fight off infected cells, and so we were able to create that. We took these infected keratinocytes, either uninfected or infected, I should say. We took their supernatant and we looked at their capacity of the supernatants to attract T cells, which are the normal responses to these viruses. And as you can see, if you infect these keratinocytes that have a wild type Diablo, they will produce a certain amount of juice that can attract T cells to them. But if you in HPV infect the keratinocytes that have the mutant protein, well, they produce less juice or less certain components of that juice, and then there's less recruitment of T cells to the keratinocytes. So we think that both of these processes, the impaired cell death in the keratinocytes in response to HPV and the impaired recruitment of T cells to infected keratinocytes, explains the very bad warts, or at least in part. Now, the other component is that this person has low CD8 positive T cells. CD8 positive T cells, for those of you who may not remember, interact with MHC class 1 on a variety of other cells, and they're supposed to be able to fight off those virally infected cells through this pathway. So we look to see if the Diablo mutation affects this process. And the short answer is yes. If you have a Diablo mutation, you express less MHC class 1 on your cell surface, as shown in the two greens here, under basal level, which is the pale green, or under stimulated level, which is the dark green, compared to any other controls, whether they be healthy controls or Crohn's disease controls, excuse me. So you're saying that, in fact, that the Diablo mutation impairs the expression of MHC class 1. It doesn't abrogate it completely. There's still some, it's just significantly decreased. And then that's an extrinsic component that's defective. We also looked, but I'll spare you the details here, is that the Diablo mutation also causes a defect within the CD8 positive T cells. So how do we use this information? Well, if we could go back to the CD8 positive T cells, we can see that those CD8 positive T cells could, in theory, respond to cells that present antigen. It's just that for some reason, related to Diablo and the MHC molecule, there's less MHC being expressed at the cell surface. So then could you capitalize on this and overwhelm the system by giving it more viral antigens? And of course, the answer is yes. You can give more viral antigens using what we call a vaccine. So by doing that, we gave him this person, Gardasil, which is an HPV vaccine, which contains HPV-like particles. And it's a series of 3 vaccines that we normally give to adolescent children. But here we gave it to him not as a prevention, but in terms of an attempt for treatment. And by about 1 or 2 months after his second dose, his warts all melted away. I should mention that his parents, because they're in the medical profession, had tried for about a decade or more all sorts of other types of therapies. Some of them cryodestructive, some of them were like, almost witchcraft from the 1950s. All of that failed. But here, by dissecting out what didn't work in this patient and why it didn't work, that is to say the Diablo mutation impacting his CD8 positive T cells, we think that we were able to circumvent that problem and cure at least his warts. And to the point now where at the beginning he couldn't play guitar because it was too painful. Now he's not only playing guitar, he's moved away to BC, which makes it very difficult to be able to follow up and get some bloods from him because he's too well. Now, I wasn't going to talk about this, but Marcel wanted me to talk about the elephant in the room, which is COVID and what we're doing about COVID. So I've had a lot of conversations with people, healthcare professionals about COVID in the last 2 years. So let me start from basics because we may have ophthalmologists in the room, right? COVID-19 is caused by a virus. Let's just start at basic facts. And that virus is called SARS-CoV-2, okay? And you can actually grade the severity of disease either using a WHO scale or something that's a little bit more convenient, which is the NIH severity scale. And we've known for the last 2.5 years or so that only some people will get severe or critical COVID and die. And those people are the older people, the elderly. Now, we never really understood why that is. And you can say, well, they're old people and therefore they have old immune systems, but that's neither scientific nor meaningful, right? Because old people have old everything and not all old people will die from COVID, even though they all have old immune systems. So I think we need to be a little bit more granular into what that means. And so using the framework that we already had in here, the RI, to understand why severe disease occurs in some people, we joined a consortium called the COVID Human Genetic Efforts to try and understand why COVID-19 severity was only seen in some people, but not in others. And we were very serendipitous, I have to say, that there is, we were able to use a biological lesson from another inborn error of immunity, something called APOSED. APOSED stands for autoimmune polyendocrinopathy candidiasis ectodermal dystrophy. So for obvious reasons, we're just going to call it APOSED. The older terminology is APS1. It's actually due to a mutation in a gene called AIRE, A-I-R-E. And what you do is, what happens in there is, among other things, you get loss of tolerance. And as a result of that, you develop autoantibodies to certain cytokines. Classically, you get autoantibodies to a cytokine called IL-17 that's been shown to be linked to their chronic mucocutaneous candidiasis. What's also been known is that these patients have autoantibodies to type 1 interferon, although viral infections are not a major feature of this disorder. But 3 patients at the start of the pandemic who were not elderly had life-threatening COVID. And that was our clue that maybe we should be looking at autoantibodies to type 1 interferon. So through multiple centers in this consortium, including here at the RI, we did that. So we set up, thanks to Lucy Roussel in my lab, a functional assay. So now, just to let you know, if this was a cell, you can actually stimulate this interferon receptor, right? This interferon receptor consists of these 2 molecules called IFNAR1, IFNAR2. There's a signaling pathway, and you can create a cell model that has an intact type 1 interferon receptor, and you can stimulate it with either interferon alpha or interferon beta, and you can use what's called a reporter, which in this case would be blue, to determine whether or not that pathway was stimulated. In parallel, ELISA that either that's coded with either interferon alpha or interferon beta. So these 2 assays allowed us to use to interrogate patients by taking their blood and looking to see if their blood was able to interfere with the interferon signaling pathway. If it didn't, you can see here through dilutions of 1 in 10 and 1 in 50, you would see blue. But if it did, then you wouldn't see blue, you'd see purple. You could also look to see if they have IgG antibodies to either interferon alpha or interferon beta. And if they have both a functional inhibition of interferon and detectable antibodies on the ELISA, well, then these people have what looks like to be an IgG neutralizing autoantibody to type 1 interferon. So that's the tool that we developed. Now, how do we interrogate that? Well, First of all, in our collaborations with others, we knew that autoantibodies at type 1 interferon were predominantly seen in the elderly population through other work. So we studied our elderly population. This is data from the from MUHC and surrounding institutions in various projects that I'm involved with. And we looked at people who are hospitalized with COVID disease. And then you can actually classify it based on either severe, which includes both the severe and critical category, or non-severe, which is anything lighter than that. And you can look to see if they're autoantibody negative or autoantibody positive. And this is our data so far. Now, we would have liked to be able to recruit more patients, but as you know, we have an REB here, so things don't move quickly. And of course, we also have patients where at the beginning of the pandemic, some of them were from CHSLDs. And so we couldn't, because they have advanced levels of care directives, we weren't even able to sample these people. But we've been able to nonetheless trudge through and collect what I think is compelling data that autoantibody positivity for type 1 interferon is strongly linked to having severe disease. Well, that's great, but what else can we use this data for? Well, in parallel, we were doing studies in elderly, non-hospitalized people, that is to say in long-term care or the CHSLDs and RPAs in CIUS Centre Sud and CIUS West. And so we looked to see that, because a lot of these people were getting vaccinated very rapidly, as we know, because of the catastrophic outcome when they were getting infected. We were only able to set up a process of, again, an REB-approved process to get into those institutions, to sample them well into the pandemic. But what we did is we looked instead of not severe disease, but breakthrough infections. And what we were able to show, again, is that there is a stronger association with breakthrough infections. So if you had autoantibody positivity, you had a stronger risk of developing a breakthrough infection. And of the 11 that are listed here, 3 had life-threatening disease. So you may say, well, that's great, but who cares, right? And you're right, who cares? How do we use this information other than to be esoteric and nerdy? Well, if you go back to the signaling pathway, we said that interferon alpha stimulates this pathway. And yes, that's true, except that these people have autoantibodies to interferon alpha, and you can confirm that because we actually developed a platform here that has an ultra-sensitive capacity to measure serum levels of interferon alpha. I mean, to the point where you're measuring in picograms per mil out of blood. So that's like attomoles, that's how low concentration, that's how ultra-sensitive it is. So there's low levels of interferon alpha. But I mentioned before that interferon alpha is only but one agonist for this pathway. You can also use interferon beta to stimulate the exact same pathway. So the question is, can you then translate that into therapy for COVID? And the answer is yes. Why not consider using interferon beta for treatment of COVID? Because some of you may say, well, that's already been done, and those failed. And you're right, they did fail. But let's look at where they were done. There was a solidarity trial where they looked at people who were in their severe or critical illness phases, or the WHO equivalent. Same thing with the ACT3 study, and same thing with another study, which I don't know what the name is, but they used favipiravir with interferon beta. All of them in the severe or critical illness phase, all of them were not good, right? No effect. So this led to the conclusion that interferon beta is useless for COVID. But if you change COVID for, necrotizing fasciitis from group A strep, and you change interferon to penicillin, you will also have the same pattern. When, you have neck fascia that's amputating away your leg, no penicillin or piptazo or meropenem is going to change that, but it doesn't mean that that antibiotic drug for that bacteria, it means that the timing is not the right time for that drug. And in fact, there's another study that looked at inhaled interferon, but in the earlier phases, the mild to moderate phase of illness. And that actually was extremely encouraging. So maybe interferon beta has a role for COVID, but maybe it's not once you have illness. Maybe it's a preemptive strategy. Maybe in somebody who is exposed and infected with COVID, and that you can prove that microbiologically, and you can then screen for them to see if they have interferon alpha antibodies. You can then give them interferon beta, because there are extremely few people who have autoantibodies to interferon beta. Really, I'm talking about a couple in about 5 or 6 consortium projects that we've looked at. So I just want to finish up with some key points. First and foremost, let's not forget that inborn errors of immunity can manifest, or at least can underlie, severe, recurrent, or recalcitrant infectious diseases, or maybe even inflammatory diseases like the Crohn's disease that I showed you. I would like us to remember that these are more present than we think, and that we are definitely missing them. And I think that by studying these patients really well, we actually have an opportunity to transform treatment for infectious diseases from a standardized approach to a personalized medicine approach. And so by studying rare infectious disease phenotypes, we can understand human immunobiology in a somewhat agnostic way, and then use that to then define and treat more common infectious disease phenotypes, as I illustrated with the candidiasis example. This is the focus of my translational research program, for which I have been very fortunate enough to have excellent mentorship along the way, including Marcel, who has supported me even when I didn't even believe in myself. And I want to remind everyone that we need to identify these patients. If there is something that's just not right and on the wards or on the consult service, let's study them. Let's identify them. Let's refer them. Let's study them, but let's study them really, really well. And lastly, let's take full advantage of the fact that our hospital connects adults to children to the research institute. And that really is a unique framework that doesn't happen very often, and certainly I think is unique, one of the few unique places in North America. And we need to take full advantage of that. And we need to be astute clinicians, and we need to be inquisitive, scientists, to be able to capitalize on the framework to transform research. I want to thank you for your time. I also want to thank a lot of people, which I won't go through, who made all of this very possible over the last decade or so. Thank you very much.
Don, a really sincere thank you. Outstanding presentation. We have over 100 people on the Zoom, and we have questions coming in. So congratulations on your terrific work, and thank you for sharing this really outstanding presentation. I'm just going to go right to questions. Sasha Bernatsky wants to know, rheumatology patients may require high-dose immunosuppressive. Some people tolerate these without recurrent infections. Some have recurrent infections Some have recurrent infections. Do you think genetic factors might explain this? And can we send these patients to you?
Sasha, yes. So let me give you an example that hits close to home, or perhaps hits close to home for you. As you know, the tyrosine kinase inhibitors we think are very smart molecular targets, but we've been unpleasantly surprised by some of the opportunistic infections that have occurred with some of them. Some of them don't even make any sense. So, for example, a BTK inhibitor we thought was a B-cell specific agent. It's actually used for B-cell specific malignancies, and yet we get things like aspergillosis, which we don't think is a B-cell related disorder. And that's because there are genetic polymorphisms. Some of them are actually more common in some demographics than others, and so that's why this rare nomenclature is a bit misleading. But there is work, and we're working with actually a friend of mine, a colleague of mine, Mihalis Leonakis at the NIH, to specifically address at least why some opportunistic infections occur with some of these targeted therapies. But the answer is yes. Now, can you send it to me? The answer is, of course. I'm a little bit swamped with other things right now with that whole, like pandemic thing circulating. But I at some point when things do settle down, I would love to discuss this further.
Fantastic. Ilana Azuelos, fantastic work and talk. Thank you. Patients with alveolar proteinosis have anti-GM-CSF antibodies or low production of these. How come they don't get spontaneous candidiasis, or are they at risk?
So that's a great question. So what I have to say for the CARD9 is that the GM-CSF response is not completely aborted. In other words, if you give other types of agonists, they will produce GM-CSF. What's defective in them is that when you get into albicans specifically, that CARD9 deficiency impairs the response to, impairs the production of GM-CSF. So that's why I think there's a selectivity in terms of their GM-CSF deficiency. On the other hand, patients with autoantibodies to GM-CSF, yes, you are absolutely right. They get alveolar proteinosis. But you know what else they get? They also get nocardiosis, but they get CNS nocardiosis. And they also get cryptococcosis of the CNS. And so I think these autoantibodies to GM-CSF patients, they are they're an overlapping phenotype because their CNS infectious process does overlap with the CNS process that we're seeing with the CAR9, but I think it's because they're not exactly the same pathway. And of course, the titer and the avidity of the autoantibodies that GM-CSF also plays a role. So I do think, though, that they are not mutually exclusive, but that they're both whispering, one in my left ear and one in my right ear, that there's a story that I'm missing.
Quick question from Ingrid Hings. If the defects is genetic, why is it only showing up in the mid-30s?
That's a fantastic question that I don't think I know the answer, other than to say is that there are neurological genetic disorders that present only in the elderly. I think what we're seeing is that when we think of genetics, we think of that it's a pediatric disease, but I don't think that genetics is pediatric. I think the pediatric onset is because the mutation is so darn deleterious that in those people that they will present in childhood. But if you have what we call hypomorphic or leaky mutations in some of those molecules, rather than being, let's say a full-fledged stop codon or something like that, then you do have this later onset manifestation. And in fact, we think about 50% of those with inborn errors of immunity are actually adults. Some of them, probably about half of those have an adult onset. But the exact timing, I think it's because we still have a lot to learn about genetics.
Fantastic. François Fabi, many nice compliments. Fantastic, true bench to bedside presentation. Question: in the context of your experiments on the SMAC Diablo XIAP axis effect on apoptosis regulation, did you perchance examine the effect of the novel mutant you identified on intrinsic induction of apoptosis, perhaps through trail or fast? Does it have the same type of response?
So that's a great question, and as François would know, right, by asking this question, because there are people who have mutations in FAS that have a very different phenotype with problematic lymph nodes, and I didn't really get into that, but we have other diseases that fall into that category. But we didn't interrogate trail, and we didn't interrogate the CD95. Well, we did interrogate the CD95, but not the trail pathway, and the responses. Perhaps I can discuss with you a little bit more offline, because I think it's a bit interesting, but I can't say that I'm a cell death expert enough to know what it means.
Okay, so I'll let you discuss with François offline, because he also has another question here. Dr. Doss wants to know, what is the role of host editing mechanisms such as ADAR, ApoBC, and which binds to the siRNA, and the RNA viral infections such as Hep C, and SARS-CoV-2?
So that's a great question. The short answer is I don't know. However, I suspect that the host editing mechanism, so far when we talk about COVID, all the talk has been on ACE2 and proteases like TMPRSS2, and that kind of doesn't hold water with newer variants as much as the older variants. But we're trying to understand why some people seem to be exposed to COVID but are never infected. Their PCRs or their rapid antigen tests have always been negative. We do serologies, they're negative. We're doing cell-mediated immune assays, they're negative, and they're clearly not getting infected. These people are resistant, which is not the same thing as people who are vaccinated, by the way, right? And we think that in some of these cohorts of resistant people, some of these host editing mechanisms may actually be playing a role in their resistance.
Marcel, I'm going to open it up. I see your camera's on and you're unmuted. You want to make a comment?
Well, I have one question, and I'd like to then make a concluding thought. Don, if I understand, these autoantibodies to interferons are happening as we get older. Why do we make these things as we get older, and what is the definition of old?
Well, I'm not going to answer that last one because I think I'm going to get in trouble. But I'm going to answer the first one. Also, the last one is a bit touchy because I say a number and then I do the math and I realize I'm close to that number. But the first question is, why do people develop autoantibodies? And what I didn't really have a chance to get into is that in our first few papers on this, over 90% of those with autoantibodies are elderly male. So that almost sounds like an X-linked recessive disorder. Now, why would that occur? And how does that occur? We're not entirely sure. Erwin Scher has some ideas, and I think we may pursue that. But the reality is nobody really can answer that question of why do these autoantibodies develop with age, and why are they predominantly occurring in males? On the other hand, we do see autoantibodies of type 1 interferon in other people, and that is to say young females who have lupus. And so I think maybe those people may give us insight as to what's going on in the elderly, but that's really, I don't have a molecular answer for that.
Thank you. I just wanted to, from the division of ID, I wanted to follow up on my introduction by apologizing for not mentioning that among the many things Don does, he's actually the service chief of the ID Hot Service, which is for the hematology, oncology, and transplant patients. And it's this level of diligence and detail that he brings to patient care that he can then bring also to the lab to understand the basis of infectious diseases. So I also want to thank Don for his service to the division.
Donna, we're going to close out in a couple of minutes, but my question to you is taking a step back and thinking of the Department of Medicine as a whole. Obviously, there is potential for lots of collaborations because everyone sees inflammation and infection and whatever whatever specialty you look at. And so I guess do we refer patients to you? When do we look out for this sort of a big picture kind of idea? Who's your team? Who do we send to you? Who do you want to see?
Well, thank you for that opportunity. First of all, unfortunately, the team that you're seeing is this. Right now, there's not much of a team. I do have a research team, but no clinical team. I think when things settle down, I would love to sit down and figure out different strategies. One thing that I've been discussing with the Department of Genetics, because I'm in that group, it would be ideal, perhaps a fantastic goal one day, to say every patient who gets admitted gets a whole exome or whole genome, because I think there's data that we're going to be able to mine to understand diseases and subtypes of diseases. But in the interim, what I'm really focusing on, just because of the amount of resources, is to say, okay, we want to focus on people who have recurrent infections. So the people who are like, oh, you're like rolling your eyes and like another requirement for a prescription for antibiotics. Yeah, those are the people that I'm interested in, but they have to be real infections, not this, non-specific ache. People who have severe infections. So, why does this person have all these furuncles everywhere? Those kind of things are interesting. Or something like a fungus growing in their brain. And then, of course, if you have opportunistic infections that you're not supposed to get, like, somebody who has pneumocystis or PJP, but they are on no immunosuppression whatsoever. Again, these are the people that we would love to study.
Fantastic. Well, on that note, thank you so much. Really, congratulations, and obviously you make us very proud. The department very proud. So continue to do this great work. Congratulations on everything you've done, and thanks for sharing all this with us today. I wish everyone a great day, and thanks for participating. I'm going to invite you, Don. There's a few comments coming into the chat. I invite you to look at the chat. There's really nice comments in there. So go ahead. Thanks, everyone. Have a great day. Bye-bye.
Thank you.